else from our crew shows up.
Clint, I'll give just a quick introduction for everybody just on kind of how to use this system. So welcome everybody. This is our second presentation on the sorghum pathology project. The previous one on the seed balls, I think, was very successfully executed. Good connectivity between 3 different continents. Video was perfect for me here, and we had good connectivity again with Maradi. Just a quick couple of housekeeping points about the Zoom technology we're using. If you use your mouse and you scroll down to the bottom of the page, you should see a bar that pops up. And when that bar pops up, you can see participants with a number 18. If you click on that, that will give you a pop-up window with all the names of everyone who is participating. And then it will also give you access to raise your hand to ask a question. If you look at the bottom of that participants pop-up window, click on raise your hand, and I'll do mine right now. My hand just was raised, and we know that you have a question. And you can ask that question when we get a break. There's also a chat feature that you can use as well to communicate a question as well. Christine will be the manager of this conference, and she's going to mute everybody's voice except for the person who's speaking. And I guess we can have Louis and Penda as well opened up in case they want to contribute something during his presentations. So we'll go ahead and we'll let the team give their talk, and then we'll open it up for discussion, questions, comments at the end.
Okay, so this is the pathology group, and we will start doing the things that we were asked to do as problems and obstacles in 2019. We had a lot of frustration among us, but my biggest one was getting Louis to Niger and Senegal. He had permission from the USDA to make the trip, but they, for some reason, said that he could not accept a travel advance. And that led to him having to pay out of his own pocket, and then eventually, months later, getting a refund for all the things. We use a thing called Concourse, and that it just took a long time. Part of the problem was that they said, well, our funds had already expired because it was after the end of the first 6 months or whatever of money that had been provided. And then the fact that he had to go on vacation time rather than, and that was because of when the trip to Niger was told that he couldn't go as a USDA person unless he had an escort. And they said we didn't give them enough time to provide an escort, and that would have been impossible to go where he needed to go and so forth. So that led to a delay in his trip because he had to get his visa from his own, not from the USDA. But it turned out in the long run, it probably wasn't so bad because it was possible then to be at the right time at the right place. And as it suggested there, getting his money back also was a major delay and problem. But we got through that, so that was good. Now then, we've also had, we were hoping to have a student maybe start in the spring semester. The first candidate just wasn't going to work because they would have to pass the ELI English exam to get in. So Dr. Pendy provided an alternative, and she had to take a course in English, which she did very successfully. And now she has actually been admitted to Texas A&M as far as admissions are concerned. But she has to get the visa through TrailNet, and our local person that's helping me with that suggested that it would probably August would be the earliest date that we could maybe expect to be have somebody ready. Hopefully by then the coronavirus problem will be over, but it's still going to put a real crimp in trying to get somebody through a PhD program. And I think Dr. Pendy will have a thing on that, which is when she would be able to enroll. She could enroll for a PhD, but if we don't have time and money to finish her, that could raise some other problems. And another problem that Senegal had was that Lewis had sent them some anthracnose differential cultivars, and apparently they didn't germinate very well because they weren't able to use those. So he has, I believe, sent them some new ones. Is that right, Lewis?
Yes.
Yeah, okay. So, and then in Niger was, I suppose based on what I sent them, their biggest complaint, and I don't think there's anybody from Niger here, but they're trying to work with University of Tilabari, or however you say that, and so that they could manage the funds that are written into the proposal for them to have activities there. And apparently it's budget, I don't know if it's still a problem or whatever, but it's taking months to get transfers of funds that can be used as they were projected in the projects. And then getting an, I didn't take out, there's two words there, an annual country workshop evaluation of year one activities and the planning of year two activities. So I don't know why that's going to be a problem now that we have this technology, but we may be able to solve that as well. And so they're wanting to be able to change the budget, I guess, in order to include plant breeding and others as part of that, part of the budget. Now then, achievements. One of the biggest thing by far has to do with Lewis's trip to both Niger and Senegal. When he went, he carried with him some equipment and supplies and so forth that were really needed there. And so microscope to Niger, supplies of fungal for growing fungi and so forth to Senegal. And as we will see on some of the more slides, the surveys were really incredibly successful. And I'll let Lewis talk about it then. And there turned out to be way more pathogens than we had anticipated as being problems. So our work has maybe expanded immensely, I'm not sure. And some of the techniques that he demonstrated for getting pure cultures and so forth seem to be working quite successfully. And Dr. Pindi can talk about that, I guess. And she added that they have a video of a survey for capitalization with interviews of stakeholders and so forth made. So that could be a very useful thing also. And I hope it will be submitted when we have to submit our next progress reports. In addition, we have managed to make some exchanges. Seeds for anthracnose differentials were sent to both countries as indicated. They didn't seem to grow very well in Senegal. Seeds from Niger, from the survey, where resistant plants and so forth were identified, have been sent. And in Senegal, we're sending them the new seeds, and they're looking at sending us the seeds from the survey made there. That would be very useful. Of course, they have to go through quarantine, but Lewis has that capacity. And Dr. Pindi informed me that she has lots of pure anthracnose isolates, which was one of our early projects that are ready to get sequencing. And now the way it's probably going to have to work, since we cannot receive live pathogens, is to have those cultures have conidia and spores, whatever, put into little tubes, microfuge tubes, and put in alcohol, which kills them. And then we can receive them and we can do the extraction, or we'll have to get some better stuff for Senegal to use for extraction, but we'll get through that somehow. The achievements still continued. The host differentials that were sent did grow in Niger, and they went ahead without, hadn't really talked about this, and did something that turned out to be very useful, and that is to test them for response to other diseases. In Senegal, as I indicated, there have purified mostly anthracnose, I think, but from infected plants. Both spent a lot of help with Lewis and the survey. and they collected seeds for resistant lines and so forth. And in Senegal right now, they're bringing in the climatic data and the information on the location so they can be part of the publication that's due to come out when all that stuff comes along. And there was really, really broad survey data collected from each country. And Louis, you want to take over on this one?
Yes, I can go ahead and take over. Yeah, what we did in Niger was to try to survey the major sorghum production regions. We missed one, but the other four are Teliberi region, the Doso region, the Tawa, and Maradi. In Niger, we were able to survey 121 sorghum production fields, mostly farmers' fields. And in this survey, we compiled the prevalence and incidence of different sorghum diseases. We are able to identify 21 different sorghum diseases, which is amazing. The pathogen system that we are supposed to work with in this project in Niger is anthracnose, grain mole, and long smut. But when you look at our table and our prevalence of what we got, anthracnose definitely was number one. It was the most prevalent disease in Niger in all regions. And in those regions as well, we were able to identify hotspots. These are areas where you have 100% anthracnose infected fields, where we can do our resistant sources for anthracnose. If we want to do it on natural infection, we have identified this hotspot for anthracnose in all these 4 different regions in Niger, which is excellent that Adamu, Isa, and Hoige will be able to conduct that. Also for long smut, although it's one of the pathosystems we need to work with, was not as prevalent as other foliar diseases like leaf blight and overleaf spot. However, there are some areas in Niger where we have hotspot, where you have actually high incidence of long smut. In those areas as well, we'll be able to screen for long smut resistance in those areas. And I'm sure we'll be successful in trying to get resistant sources for this particular disease. The third pathosystem we need to work on is grain mole. Based on the time that we did the survey, most of the fields were not mature enough. So we didn't have a clear picture on how prevalent grain mole is in Niger. But the other scientists in Niger were told to go and resurvey those fields later in the growing season when the plants are mature, so that at least we can have a clear picture on grain mole on sorghum. The other thing that we did also was trying to, at least after this survey, ask those scientists to continue to do survey on another region of Niger where sorghum is really important. I think that region is number 1. So I asked them that they need to actually go to that area as well and survey the different diseases. I wouldn't be surprised that they'll find similar results as we found in this previous survey that we did last year. The next slide.
Yeah, this is
Senegal. In Senegal, we surveyed more fields in Senegal than in Niger. We surveyed 206 fields in Senegal. In Senegal, the responsibility is to look for resistant sources to anthracnose and grain mold. As you can see in Senegal, when we look at the prevalence and incidence of disease in Senegal, we found only about 15 different sorghum diseases that were prevalent. Leaf blight was number 1, and anthracnose was number 2. However, we are not responsible for doing any evaluation on leaf blight. But based on my discussion with the other scientists down there, I told, especially with Cyril, that they actually need to also look at leaf blight in their evaluations. Because if you have resistant to anthracnose but don't have resistant to leaf blight, they will still impact sorghum yield in Senegal. The other thing that I observed in Senegal was that Indra and Sarah, they've developed some improved varieties. But in those improved varieties, the disease that's most prevalent is not anthracnose. It seems that their improved varieties have good anthracnose resistance. However, they are lacking in resistance to leaf blight and also to sooty stripe, which means in their evaluation for both the exotic lines and the improved Senegalese line, they need to also include screening for leaf blight and also for sooty stripe, since the majority of those improved varieties are actually resistant to anthracnose. I found very, very, very few infected improved sorghum lines, Senegalese sorghum lines in Senegal when we did the survey, which means they will have to also include evaluation for leaf blight and sooty stripe. When the table that we'll show you will show the prevalence and incidence of the different diseases. In Senegal as well, we were able to identify hotspots. So if you want resistance to anthracnose, there are areas in Senegal that we've identified where they can actually put the materials and evaluate them for resistance to anthracnose. Here again in Senegal, the time that we conducted the survey, the plants were not that mature. We didn't have that many mature fields. So we can't say that much about grain mold, which is one of the responsibilities that we need to look at, resistance to grain mold. However, the few fields that were mature with their improved varieties, those improved varieties were susceptible to grain mold, which was surprising. They were very clean for most of the foliar diseases, including anthracnose. But when it came to grain mold, most of those improved variety fields planted with the improved varieties were susceptible to grain mold. Okay, Dr. Mcgill, you can move on to the next.
Well, before we move on, I just wanted to remark that if you see all the spots they went, Lewis was there for 3 weeks, and I think he lost 30 pounds. I don't know how much his collaborators lost with him, because they were with him every step of the way. And so this is really a remarkable amount of work and remarkable advance, I think, for this particular project. So now this is when Lewis got back and regained a little weight, I guess, he went right to work on the publications. And the first publication that came out had to do with the use of these inbred cultivars for anthracnose resistance that are host differentials being tested. And some other interesting results of that showed that SC 748-5, which is our standard for anthracnose resistance, is susceptible to several other diseases, at least in Niger, and also identified a good potential source of resistance to leaf blight. And the second publication that's already out, I think now, it says in press, but I believe it's been released, talks about the stuff that he saw in Niger with the 21 different diseases and the relative frequencies and so forth. And so those papers will be available for everyone to read. And I need to remind Lewis that if they're not in open source, we need to send the data, survey data and so forth, into one of those open resource places that we talked about last week. Here are some of the just incredible pictures of the diseases that are in one of the publications. And upper left, anthracnose, and anthracnose in the midrib, which seems to be a different resistance mechanism than the leaf blade. And then you can see if you see these things sticking out up there, those are the long smut, and that's why it's called long smut, I guess. And then leaf blight and zonate leaf spot, which is pretty easy to detect. Rough leaf spot, which I always confuse with anthracnose, but Lewis can look at it in a second and tell the difference. Head smut, which we have more problem maybe here than they do. And then finally the oval leaf spot. I don't know if there's really good information on the relative scaling and so forth of how much it has to do with loss of grain, but I'm sure anytime you lose some of your good leaf surface, you're losing potential energy. So there's probably a cost for these diseases. Now, going back to our original objectives in our proposal, I think the first one about making a collection of sorghum varieties, pathogens, and so forth, I think that's been handled remarkably well. I think they have collected samples of the sorghums and the varieties and so forth, and know where they came from and which ones are potential resistance and so forth. The second one was about the purification of isolates, and now Dr. Pendy has done that, even though she had to take some time off to have her new child, and I hope everything's well with that. But anyway, she tells me that they have lots of samples now, and we'll have to figure out how we're going to get the DNA sequencing, which was part of that particular project that we suggested to compare the differences and so forth, especially if we can get race identification involved. And then to identify the species that are causing grain mold. Well, as you heard Lewis say, that didn't work out so well during that trip now, but maybe they'll be able to collect those kind of data on what it is with grain mold and how prevalent it is and so forth. But that one we haven't got to entirely yet, but there are more. Lewis talked a little bit about the long smut problem. And yes, there were areas where it was pretty high, and they have identified some hope. I think they probably have hosts identified that may be resistant, maybe haven't been verified, but they can at least go to those hotspots now and test them. And then these USDA collection of sorghum germplasm from Niger and Senegal, those seeds have been sent to us. Senegal, I believe, we have completed. Mm-hmm. And so we know which ones are resistant to anthracnose and which ones aren't. So Lewis, you want to add anything to that?
Yes. We evaluated, I think, 167 Senegalese lines that are maintained by the USDA germplasm collection in Georgia. So those lines were evaluated for anthracnose resistance in the greenhouse. We've already compiled the data, is completed. Now what we need to do is just analyze it and probably publish the information as well. But with Senegal, this is not the first time. We also evaluated those similar lines in Puerto Rico and found a large number of resistant sources to anthracnose. Okay. So the lines that we identify, will send them to at least give the information to Penda and Cyril, let them evaluate them in Senegal as well to look whether they are resistant to the pathotypes that exist in Senegal. But the information for those 167 Senegalese lines that we maintain within the USDA collection, most of them were resistant to anthracnose, to the pathotypes that we have here in Texas and also in Puerto Rico.
And fortunately for us, many now of all the Niger and Senegal cultivars have, courtesy of Jeff Morris, been sequenced. And so if we can, maybe we can use those as a pattern to see potential resistance that shows up in a Manhattan plot when we look at the sequences of the resistant versus susceptible. That's not the first idea of this project, but it pops right to mind when we have all this information on which ones are resistant and susceptible. If the sequences are there, we can see if there's a pattern of regions of chromosomes that seem to be associated with resistance or susceptibility. Now then, the planning for this year is, first of all, to get that Senegal survey published. We've already talked about a lot of the things that we'll have to talk about how we're going to do it. I don't know if we have to make a whole lot of changes in our goals and whatever, but be able to use the hotspots for testing other cultivars than are typically grown there to see if they're resistant or susceptible to those particular diseases. Identifying resistant cultivars at various locations to the most critical diseases. I think that should be something that we should try to manage since we have all those new data. And to make the crosses to create cultivars. If you have one that's resistant to one disease and another cultivar resistant to the other, it'd probably be a good idea to start making the crosses to try to stack those resistance genes together. And then use information for more farmer informational and training sessions. Obviously, if you know that you shouldn't be planting a certain variety a certain place, it might help the farmers to know that too. So anyway, now then we have to do something about resolving the status of the students that we were hoping to hire. I will say that it was a good thing we didn't have somebody this spring semester, because the funds weren't here yet that we had planned to use for starting a student then, so we wouldn't have been, we'd have been a problem there too. But in Senegal, they didn't get new students, but they did pay 2 former students to do a lot of the work on the pathogens, and they hired someone who cleaned the lab apparently. And now verified pathogen identification by ITSs. Well, this was something we thought would be good for the graduate student when they came. If we can get DNA from things, one of the quickest things to do as far as making a neat little project is to use primers that flank the ribosomal ITS internal transcribe spacers, because they're, when you sequence those, they're very, very species specific, and they can be used as identifiers. And so that was something, again, we will have to get the killed DNA samples, or killed either DNA samples or killed spores from Niger and Senegal in order to do that. And then in Senegal, they would still like to have some other equipment directly. Mame, do you want to say something about this? What you need? It looks like she's shut off. So
yes, she's having difficulties, he said.
Yeah. So anyway, don't need a PCR machine for doing DNA extraction, but you probably would could do that work there if they had one, that ITS stuff, and because sequencing can be hired out anywhere now. We have a local part of our ag experiment station. There is a sequencing lab, but they're pretty expensive compared to some of the others. So anyway, this can be done. For the objectives that we had listed for the first year, I think, again, we did pretty well. Purify, expand pathogen cultures in Senegal and Niger. Identified local resistant cultivars. I think that happened pretty well, possibly at least. It have to be verified. And verify pathogenicity of the things, that the isolates, to see which ones attack which cultivars and so forth. That's still part of the objective in year 2. And test sorghum anthracnose gene lines with local isolates. Well, we've done that already for Senegal, so maybe for the Niger we can do that as well. And then obtain and sequence DNA. That's still something for this year that we were hoping to do, and I think we'll manage to get some of it. And as I mentioned before, Jock Morris has already sequenced many of these things, so we won't have near as much expense and work as we thought we would have. And then in objective 2, our thing was to look at pathogen diversity, and we're well on our way with all the isolates that PENDA has collected. For the races, again, we've already done the Senegal. So that's a year 2 that we're ahead in. The verification of the species, now we have not done that, but as soon as we have DNA, Texas, that's T, that's us, we can do that. And the rad sequencing of the pathogen can make phylogenetic and geo-based comparisons among the races and strains and DNA and so forth. I mention this because some time ago I was on a project with pearl millet pathogen, and it turned out that they did all the breeding for resistance in India, sent them to Africa, various countries, not these 2, but, and they said they were all susceptible. Well, that's more or less what you'd expect. And it turned out they were really 2 quite different sets of isolates from the 2 different countries. So they really needed to have different genes involved in those particular cases. And then starting the development of populations for gene tagging. In Senegal, Mami tells me that they have already got an F3 between SC748-5 and some local varieties that if we get those segregating out and can test them out, well, then we would have a way to really get into that tagging. Now, there has been a gene tagged in that one already, so maybe it's the one and could be followed as well. So anyway, these things are, I think, still rather realistic for our second year of trying to get things advanced. And then we had objectives 4 to 6 that were all for the later part of the project, the years 3, 4, and 5, or whatever we started with, to try to get tags, good tags for specific resistance genes that could be identified with simple PCR tests. So as far as I can see, we're making some kind of progress, and mostly thanks to Lewis and our collaborators in Africa, not to me, but we're making some progress. And now here's the contingencies and COVID-19 strategies, based on the fact that, according to Dr. Dalton, it's almost certain that there will not be any no-cost extension, which maybe I keep thinking if COVID lasts a long time, that may get overruled. I just was on a project, a 5-year grant with Johnson grass and sorghum, where we had got the money so late the first year that really in the 5th year we were spending 4th year's money, and we were able to actually get that as a no-cost extension. And then we just found out last week we can have another year to finish because there's been a big delay with USDA moving and so forth. So anyway, we did get an extension that might not have normally been possible at all because we'd already used the no cost to cover the 5th year. And anyway, so our student programs in all locations may require some alteration if this Kumba Fall, who's the student that we've identified and admitted, can't get here in time to do a PhD. What Dr. Penda has suggested is that maybe she should get a PhD there and could come here and spend time as an associate or something to do the molecular work, and because the money will run out probably before a PhD could be a realistic outcome. And so the other thing that affects all of us is because of this COVID thing, it may be hard to have meetings with farmer groups and so forth of any size, and it may be hard to even travel. So we hope this all gets over in a short period of time. I don't imagine many of the local farmers have access to Zoom, and that would be one way maybe, but you can spread the word among the researchers, but it's going to be hard to get it down to the local level if you can't travel and if you can't have meetings. So I think that's all that we had on our thing here, and I'll stop sharing the screen that way. And it looks like Lewis is on the screen, so on my screen anyway. And you guys can take over if you want.
Sure, let's start first, Louis and Dr. Penda, if you have any comments to add.
Yeah, I think the only thing that I can add right now is that the lines that they are sending us from Africa, we can test them for our local isolates here. However, we are not sure whether the same lines will respond the same way in Africa. The other thing is also to look at the different pathotypes, the different races of anthracnose in particular. They will have to use at least a chamber, a greenhouse, or a glass house in order to do the different races of anthracnose. Because if they put them so close together, they will have interference. They got these single isolates, but now they will have to, at least as I said, if they want to look at the different races on anthracnose in Senegal, they'll have to conduct that in a greenhouse.
Now, in that case, it would be especially good to have the DNA analysis so we would know which ones are most different and so forth.
Then use from those clusters, probably that would be easier. if we can do the DNA analysis, have a dendrogram or phylogram, then from each cluster, they can identify a representative isolate, then do it in a greenhouse. Because I don't know how much greenhouse space Penda has in Bombay or in Chess.
Yeah.
So that way it will be easier. You don't want to use all the 200 and something isolates that you have. That's too much.
No. Yeah. I agree with Prom. So the best way is to do the sequencing, and if you have like 2 groups of isolates, we use some strands. If you have 5, 6, it's okay, and we can do the pathogeny test. I have a greenhouse in Bombay, but actually we need to install the tables. It's covered, but we need to install the tables before we start the work. I can do it with the project.
Okay.
Great. Have a large.
Yes, I think that will work. That's the only way, because here I can afford to do 100 different isolates because I have several greenhouses here at the USDA and also on campus where I can do the pathogenicity test. But in your cases, I would rather do the DNA sequencing first, then use those, as I said, representative isolates from those clusters to look at the different races in different regions of Senegal that way. That would be much easier. For long smut, we don't have an established set of differentials for Niger. It may not affect Senegal that much because I didn't see that much long smut when we did the survey in Senegal. However, in Niger, in order to identify the different pathotypes, the different races of long smut, we actually need to look at our some of the lines that we have in our sorghum differentials, as well as some of the different local lines in Niger to compile a set of differentials to enable us to determine the different races of long smut in Niger. We've collected, in fact, in Niger, we collected different isolates of long smut in different regions of the country. So they have those stored in Teleberry, as well as anthracnose. They haven't isolated the anthracnose yet. We haven't gotten any information from them that they've done some single sporing of the anthracnose isolates in Niger, but they also need to do that as well.
Okay, so let's open up the floor to questions then. And the first one was submitted via the chat box by Peter Matlin. And so his question is, how do the survey results on the prevalence of major sorghum diseases compare with the large number of regional and national field surveys conducted over the last several decades in programs coordinated by ICRISAT and SAVGRAD and published in refereed publications, as well as annual reports of ISRA, INRAN, ICRISAT, and SAVGRAD? And what do those differences from those earlier results tell us?
Oh, we didn't have any information on that. I spoke with the scientists in Senegal. Has this ever been done? Have they ever done an extensive survey of sorghum diseases in Senegal? Do we have any information? Do we have any documentation?
No.
The response that I got was, I don't think in Niger, they are sure that there has never been an extensive survey of sorghum diseases. Yeah. I don't know in Senegal, but when I talk to them, I'm not sure whether an extensive survey like this one that we did has ever been done in Senegal by either ICRISAT or the USDA.
No, in Senegal, it is the first time we did this survey. We didn't have any information on diseases before. Yes. That's why we do it in this project. We didn't have any information. This is the first time we did it.
Exactly. In Niger is the same thing. This is the first extensive survey of sorghum diseases in farmers' field in Niger. So there is no documentation. So the surveys that we've done for both countries will be the first documented survey of sorghum diseases in both countries.
Yeah.
Okay, other questions for the team? Or Peter, do you want to follow up, or are you okay with that response?
Just a very quick follow-up. I know that you can find such survey data from some of the early annual reports that ICRISAT published of the sorghum network field surveys that were done. Uh, so it may be having to look at some of the gray literature. I know also if you look in some of the CABI publications, you will find results of field surveys of the prevalence of the major sorghum diseases in the Sahelian region. I would have to, dig into it right now to find out exactly which regions and how much the coverage was in both Senegal and Enron. But those reports exist. Again, it may be in the gray literature or in annual reports. But also look at CABI, because I just did a quick search on my computer and found a number of CABI publications that had them.
They had surveys of how many fields?
Well, again, you would have to look at that. But the point is that the surveys were done. And the interesting thing, I think, is to determine whether or not there have been changes in the prevalence, and then trying to interpret what does that tell us.
Okay, maybe later we can see these reports and see the differences.
Yes.
Yeah, because I didn't know about these reports before. And I am sure in 10 decades, maybe we will have changes by the climate change and things like that.
True.
We will have new pathotypes, something like that.
True.
Yeah.
Because they've injected a lot of new varieties in Senegal.
New variety, new varieties, new realities, environment, the condition.
The older land races also have been replaced by newer.
New, ameliorated, new,
I'm sure.
new lines.
Okay, Mampenda. Hello.
Yeah.
Okay, what I can add is most of the previous survey did in Senegal was only located in four localities: Sefa, Sinthioumalem, Bambey, and Daru Pakachar. So what I can add is a survey like we did with PROM. I believe that it never been.
It is the
done.
first time. Yeah.
The first time. The previous survey has been located in a few locations, but not widely as you did in this survey.
Yeah, for sure.
Good information to follow up on. We have a question from Ghislaine Kanfani. In Senegal, do you have a greenhouse where the screening for anthracnose could be done? I guess you need to control relative humidity and temperature.
Okay, so yeah, I can answer.
Set up a misting system in the greenhouse. You can hand mist them on a daily basis as well, so that should work also.
So we can try to have the conditions like irrigating, something like that. We cannot control the temperature. We don't have this is in the greenhouse.
Yeah, you just mist the
But we can control the humidity with the irrigation. But for the temperature, we cannot control. We don't have stuff for this.
There's a question in, sorry, somebody else going to respond there? Let's move on to the next question. There's a question from Bettina. She asks a two-part question. Since this is a new project, could the project team be presented? So Louis, Clint, do you want to go ahead and introduce everybody?
Okay.
Well, I'll just introduce myself and tell who the others are. But I'm Clint Mcgill. I'm a professor of genetics at Texas A&M University, and I'll switch it to Louis.
Yeah, I'm Louis Prom. I'm a sorghum pathologist for the United States Department of Agriculture, based in College Station, Texas.
Okay, I'm Mampenda. I'm from Senegal, Israel. I'm based in Bambay, and I am responsible of the plant pathology lab in Bambay.
Yeah. And we have other collaborators.
I'm Cyril Jata. I'm a sorghum based at Bambay, Senegal.
Your video.
I'm glad to hear you're here. and
I can see you, Cyril.
No, I shut my videos. I can. Yes.
Okay.
There he is.
Just one time, yeah.
Mhm.
And the co-PI in Niger is Adama Haoudi, if I can say that
Yes.
right.
Mhm.
And then there are several other participants in both places, but that's who we confer with most often.
Okay, a couple more questions from
Thanks.
Bettina as well. Could you share a map of the disease hotspots identified in the survey? So that would be very helpful. You just presented the maps of where the surveys were done, but as you start putting the prevalence in there, those hotspots would be interesting to look at. And then the third question is, are there any plans to consider farmer preferences, farmer preferred traits in the plant resistance breeding work?
Yes, the hotspots, actually, we know specifically we can, for Niger and Senegal, we'll be able to put a reference point in the map to the exact fields because we have the coordinates and everything. For Teleberry, we had one in Fera Mauri. In Doso, for anthracnose, we had two hotspots. We have one in Benguai and the other one was in Gaia. So those ones we can put definitely in the map. We have the specific farmers field and the farmers contact information as well, where they can actually evaluate their collections down there for anthracnose resistant. Tawa, we have two areas in Tawa, in Niger, and in Maradi, we have one area. So for these are for anthracnose. We also have other hotspots for other diseases during the survey that we can put in a map. In the the article that we will be published in Plant Pathology Journal, we put the information in there where the hotspots are located and the specific fields and their coordinates. So people can actually go exactly to the fields where they can negotiate with the farmer to put their materials if they are looking for anthracnose resistant head smut, long spot resistance or any other foliar disease such as leaf blight, zonate, or rough leaf spot. So we have these hotspots, we have their specific names, and we also have the field numbers and their coordinates.
Okay, so in Senegal, so we consider farmer preferences when we were doing the service. That's why we did the video. In the deep video, we have an inquiry where we ask producers about the disease. If they know, you show them the photos of the diseases and ask them if they know the disease, how do they do when they see the disease, something like that, so that, and ask them about the project, what they want about the project, what they are waiting for, something like that, so that after 2 or 3 years, now we are going back and ask them again at what level they are, if they have new thing about this project, if this project brings something for them, something like that. So that's why we did an inquiry also when we were doing the survey in Senegal.
Yes, you are right.
Is a video, and in the video, in the video you can have all this information, all the background about the producers, what they think about diseases in Senegal. And also hotspot, we can identify hotspot. But as Tom said, we need to do a publication, and in the publication you will have all these things inside.
Yes, true, yes. Yes, as I said, in each farmer's field during the survey, the most interesting part, the only way you can transfer this technology is to interact with the farmers, actually. So what we did during the survey is that in places we met up with the farmers, we tell them what the different diseases. We also emphasize the management strategies that they can use in order to reduce the impact of the diseases on yield. In Senegal, fortunately in Senegal, we were able to videotape them. In Niger, we didn't do any videotape, but we were able to interact with a lot of farmers. Not only did we explain to them about the diseases that we are working with and the diseases that we see in the field, we also took their contact information. You know, we have the contact information with us as well. So which is very good. So this is the only way that it will be easy to transfer the technology from this project into actual farmers' practices in their respective countries.
Okay, a good follow-up question from Njito. Is there any recommendations on how to adjust the sorghum breeding program, or do you have any recommendations on how this should affect the sorghum breeding program?
To me, immensely, because with Cyril, who is a sorghum breeder, he was with me during the survey, and I've explained to him that based on what they've developed and released in Senegal, the improved varieties, he identified them during the survey and said, this is an improved variety, that's an improved variety from their institutions. I told him that you look at your improved varieties, they are actually, you have good source of resistance to anthracnose. However, they are really susceptible to leaf blight and sooty stripe, which means in addition to looking for anthracnose resistance, they should pay attention to leaf blight and sooty stripe for the improved varieties that they are releasing to these farmers, because some fields you have 100% incident. Every plant has leaf blight. In other fields, it looks like some of these improved varieties, every plant has sooty stripe. So what do you do? So you have to prioritize and also include in this project. And I know that we are not supposed to address these two pathosystems, leaf blight and sooty stripe. But since most of their improved varieties are highly susceptible to leaf blight and sooty stripe, they have to include that during their breeding program to look for resistant sources to those two diseases, because it's impacting here. I can see it from the fields that I visited, that actually these two diseases are impacting here.
I think just a follow-up point on from me, that I think you're correct to some extent on that point, Louis, but if these diseases are happening in a very small locality, and this is back to Bettina's point, do we have an understanding of hotspots and where these diseases are located? If you're seeing it being highly geographically concentrated in one area, they may not be sufficient reason for a blanket incorporation of all this disease resistance into every single variety, or more specifically to maybe you want to target it to
for the improved, yes, that's what I'm saying. For the improved varieties that I've seen in different fields, they were uniformly susceptible to leaf blight and sooty stripe, which means it's a wide geography, it's the entire country, practically, where they grow sorghum. These varieties were susceptible to leaf blight and sooty stripe. And the breeder was there to see it himself. Cyril was there.
Yeah,
it's not isolated. It's everywhere.
I confirm what Prom said. Most of our improved variety were susceptible to leaf blight and sooty, but have a good behavior for anthracnose. Anthracnose is not a problem for our improved varieties. So another thing I can add, during our survey and last week also, we collect seeds around the countries. So during the coming rainy season, we have to screen all of the accession for most of the diseases. This study will enable us to identify new sources of resistance. I'm talking about local varieties that have resistance for a particular diseases. This is also something that we can use in our breeding program to improve those that have been susceptible. An example, as we said, as Prom said, most of our improved variety are susceptible to leaf blight and sooty. So if we identify local varieties that are resistant, we can use this variety to improve the new release varieties. Another thing I can add for Bettina questions, in my program, I'm implicated in 2 or 3 small projects. So sometimes what I'm doing is to connect all phytopathology project, the Simil Sawajan project, and the digestibility project. I'm connecting it to answer my questions. So, for example, for the farmers' needs in the Simil Sawajan project, we are addressing this question to have all the requirement, what the farmers' preference and need for sorghum varieties. So if I connect farmers' preference to the breeding program where we can correctly answer or deliver technology that require that answer farmers' needs. Hello.
Okay, good, great. No, thank you very much. And I think along the lines, it's something to think about. We've had good discussion on this. And I know this was not part of the original design, but Peter Matlin asked, how will the team assess the magnitude and frequency of the impacts of different diseases on sorghum grain yields in each country and agroecological zone in order to prioritize your work going forward? And so I think what he's really looking for, and his perspective as an economist, is one, we can't breed for everything. So what are the highest disease priorities that you may want to focus on? And have you thought through how would you go about making those recommendations? And Cyril, I think probably your expert opinion on this is also very useful.
Oh, is that directed to me?
I think it's directed to the team.
For all the team, yeah.
Oh, okay, that's great. How do we make a decision?
I know.
Prioritize.
Okay.
How do we make a decision or prioritize which pathosystem to work on? Which of these diseases cause the most significant yield? This is why you do survey. We did a survey.
Yeah.
We looked at the majority of fields, farmers' fields, production fields, and look at the prevalence of these diseases. The severity for most of these diseases was not looked at. But as a pathologist, once I entered the field, I was able to look at the severity and what type of yield that farmer may most likely encounter. And we can see it, as I said, this hotspot, the reason why we identify this hotspot is because in those areas, the field, we can talk about the environment or anything of that sort. I can understand talking about, well, the environment, probably when you look at the geoecological stuff. Yes, obviously, but that doesn't mean that the disease is not present in other regions. The reason why you don't see epidemic every year in any country, sometimes it's the environment. That doesn't mean that the pathogen is not there, it's present. What about just in case you have a change in pattern of the weather, where you have the rains coming either early or late, and you have your materials in those countries? Looking at the prevalence at least gives you a roadmap as to what diseases you have to concentrate on. You cannot say, okay, the environment is not conducive. Looking at the disease triangle as a pathologist, I always go back to the disease tetrahedron or the disease triangle. The reason why I don't see anthracnose infection in College Station every year is because the environment is not conducive every year, even when I inoculate the plants to have an epidemic proportion of the disease. So looking at prevalent, I'll say, okay, well, I'm here. Anthracnose is the most prevalent disease. So where do I put my resources on? Trying to identify sources of resistance to anthracnose. If I say in this region is leaf blight, then I have to concentrate on leaf blight. So that's why we did this survey. This gives them a roadmap for the future, not just for this project, but for the future to say in the next 5, 10 years and say, well, we did a survey 5 years ago, and this is what we found. We can also do another survey like I normally do in Texas. I do a survey every year or every other year and say, okay, well, did the prevalence change? What happened? Do we have new varieties, or do we now have resistant sources to the pathosystem that we've been working with? Now it's less prevalent, let's go to the next one. So you prioritize it based on what the pathologist sees on an annual basis or on every 4 or 5 years. Yeah. We can't constantly say that, well, go ahead, Penda.
Yeah, my idea. So when we were writing the project, we were focused on anthracnose and grain mold in Senegal. But after the survey, we know that anthracnose is not well important in our varieties. So now my idea is, can we focus on leaf blight and anthracnose and also grain mold, all the 3 diseases in Senegal, or we need to focus only on anthracnose and grain mold as we did when we were writing the project. It's a questions too.
Okay,
Because
well,
me also, I am a little bit confused with this, because after the survey, I know leaf blight is a reality for us. And when we were writing the project, we didn't focus on leaf blight. We only focused on anthracnose and grain mold in Senegal.
Yeah, my thinking is what you need to do is,
yeah,
when you are evaluating for your anthracnose resistance, you guys are there already. You know where the hotspots are. Why don't you use the same varieties in those other locations as well? Or you can do a multiple disease resistant project. You can do multiple screening for these diseases. Not only do you inoculate the plants with anthracnose, but you can inoculate them with other pathogens as well. And look at one that seem to be this thing. So you can do multiple disease resistant screening anywhere that you, anytime that you guys want, because they've already set their priority that this is what you guys want us to work on. But generally, as a pathologist, I would have rather done the survey first before deciding what pathosystem you guys needed to work on. That's the way that I would have approached it. But as I said, it's already gone. We've already started it.
Let me go ahead and just address that point. Prior to even writing the call for the proposal of this project, we surveyed the sorghum system in West Africa, and these are breeders and the research team leaders in sorghum, not only in Senegal and Niger, but across the region. And these were the diseases that was prioritized. Right. So that's why this occurred this way, Louis. It was not just a haphazard idea, as perhaps you're suggesting. Now, more specifically to Dr. Penda's point, well, this is a very interesting finding. If this is not as a high priority as was suggested by that initial survey, then I think we should consider reorienting the program. The third point that I'd like to bring more generally in terms of prioritization, it's not just the prevalence of the disease, but it's the damage, the yield damage caused by that disease on that yield, combined with the probability that we can make some progress on reducing that yield loss.
Yes.
And where we have the highest potential to solve a yield loss problem, combined with prevalence, then all those three elements would combine to be a high priority, as opposed to, there's a lot of disease, the yield loss is very minimal. That's a different type of potential benefit to the producers that could occur. So I think this whole thought needs to be, I think this whole process of evaluation of priorities, I think the process could be developed to be a little bit more sophisticated, because Njito, as the leader of CIRAS, is asking, do we need to revisit our breeding profile by prioritizing leaf blight? I don't think we have enough information at this point, Njito, to say that, but certainly new questions are evolving, and certainly this survey is creating refined sets of questions that we need to take into consideration. So I think there's plenty more to discuss on this topic.
Yeah, and I don't think anybody right now is in a position to say what the yield losses are, especially on an annual basis. So it's a huge problem to measure those kind of things. So I don't think we're going to do that, but we are scheduled in the project to have, not this year, but next year, Lewis go back and do another survey. So maybe we'll find out something about how they're, it might be a whole different year, of course, but find out how prevalent they stay. So that's something that will happen.
Great. I mean, and Njito, I think this is a great topic for you to keep on the agenda with your team as you're discussing these issues. Do we need to rethink these priorities that we're breeding for based upon less, the more comprehensive information we've now received? Dr. Penda and everyone involved in this discussion, I think you're in a great position to pull the team together and talk about this and try to be objective in setting your priorities.
Okay, that's fine.
Other questions for the team?
There is a thing on the chat asking if there's a known source of leaf blight resistance in the U.S. germplasm. And I can't answer that question right now. I could
Well,
probably find it in a hurry.
it's not, as I said, it's not a significant disease for us in the United States. We just don't encounter leaf blight at an epidemic proportion in any regions of the US, even in Puerto Rico, which is a tropical country where we do some of our evaluation. But do we have a resistant source for leaf blight in the US collection? Of course, we have over 40,000. If I go there today, I'll be able to have resistant sources to leaf blight from isolates from the US and also isolates from Puerto Rico. Yes, there are resistant sources, but that's not a priority. We just don't do that in the US. We just don't look at it as an important disease that we have to work on. I know the federal government doesn't see it that way. It's not just an important disease here in the US, but there are sources of resistance. We have enough germplasm to be able to go and get resistant sources to leaf blight.
And I guess the other question, which is unknown,
Okay.
there is.
I add something also in my program also. There is Surenos that have a good resistance for leaf blight also.
Soreno.
Okay.
Okay.
Good question. From the audience, do we have any more questions? External Advisory Board members, any additional questions? Okay, so to the team, a couple of points from the management entity. One of your early slides that you put in, Clint, was that you were having difficulties with contracts between INRAN and University of Tilbury. You need to bring that to our attention as soon as you have problems. That's our role, to try to troubleshoot any headaches you might have. So in administration of any funds, please bring it to our attention sooner rather than later. This is the first we've heard of it.
Oh, I'm sorry that Niger wasn't on the line.
Yeah. Well, let's follow up. Let's try to follow up with a conversation in a way. We can do a WhatsApp discussion with them, but we'll certainly follow up on that. Secondly, again, you have an opportunity to think critically about what you've learned. And as I've mentioned in the previous call, this is all about adaptive management, our program. So as you're learning more, if what was conceived as an original objective, which you find to be of lower importance, let's revisit it, let's discuss it, let's have a strong argument of why we pivot or shift our attentions. Okay. So certainly we're trying to solve the problems of the highest priorities, not just check off boxes that we're doing what was originally stated. This is science, and let's continue to learn from, we went on with the initial hypothesis. That's what was set up, that anthracnose was the most important. Ill-defined support for that hypothesis. So let's learn and pivot.
Thank you.
Anybody who was not present at the first meeting, these conversations are being recorded, so we will post them online if you'd like to pull it down and take a look or share it amongst other colleagues. Again, please contact Christine if you're not able to download, and we can send a USB with the presentations to you for wider distribution. In terms of other, just a second, I have one more question coming in. Let's finish my thought and then I'll turn to that. I'll leave it at there. Bettina has one last question. Which of the diseases has the largest negative impact on food safety and mycotoxins?
And grain mold.
Grain mold, okay.
Yes,
that's
because
most of the, that's a myriad of pathogens.
Yes, that are capable of producing, capable of producing mycotoxins, but based on what type of fusarium or what type of alternaria or other species of fungi that are associated with grain mold. It's a very, very difficult subject, and it's a very complex disease. When we look at grain mold in its totality, I always say you have a synergistic effect. It's not caused by one pathogen. It's actually associated with pathogen in over 40 genera, which means it's very, very difficult. But the ones that we tested in the US, the ones that we identify here in the US, the fusarium, the tapsinum, the semitectum, the chlamydosporium, etc., they have the capacity, and also the alternaria, the cupular. Some of these have the capacity to produce mycotoxins. So they are actually, they impact not only yield, but also quality
Yeah. That's why when we do the second trip for grain collection in Senegal, we also collect it in the market to see
Yes.
the fungi, the most frequent fungi we'll have. And after, we will see the hotspot and see which is frequent, more frequent, and if it is mycotoxin products produces something like that.
Yeah, most of, as I said, most of the fusarium associated with grain mold have the capacity to produce mycotoxin. So there is a concern in food safety, definitely.
Yes. Thank you.
Good, excellent. All right, I don't know if there's any additional questions. If not, I want to thank Dr. Penda for being available and connecting well. Cyril, thank you very much for being available to contribute. This has been so much of a richer discussion with the two of you involved, with Clint and Louis, who has easy access, and unfortunately we didn't get the Nigerian counterparts involved, and we'll have to do our best to try to connect with them at another point in time to make sure that we better understand the situation, any concerns they have there. So I'd like to stop then this presentation and